Welcome to our conceptual summary, and today we will be talking about carbohydrates. Carbohydrates basically is just a molecule of carbon that is attached to a molecule of water. So the general formula for carbohydrates is actually equal to C subscript N, then the molecule of water subscript N. For example, if our N is equal to 6, therefore, we will have C6, H12, and O6. This is the molecule for glucose. Glucose is a sample or an example of carbohydrates. The different types of carbohydrates. The different types of carbohydrates are the following. First, the monosaccharide. Monosaccharides are sugars or carbohydrates that only has one sugar molecule. The example to this are the following: glucose, fructose, and galactose. The other type of carbohydrate is called a disaccharide. Disaccharides have two sugar molecules, and the examples are sucrose, lactose, and maltose. While the third type of carbohydrate is called an oligosaccharide. Oligosaccharides have 2 to 10 sugar molecules. The examples are raffinose and stachyose. While the fourth type of carbohydrate is called a polysaccharide. Polysaccharides have 10 or more sugar molecules. The example to these are the following: starch, glycogen, and cellulose. The digestion and absorption of carbohydrates. Carbohydrates, an example to this is a starch. A starch is broken down by an alpha amylase into a maltose. That is just one example wherein a carbohydrate can be broken down by means of an enzyme. So generally, carbohydrates are digested in our mouth. Chemical digestion is actually caused by the salivary enzymes. And an example to this salivary enzyme is called the amylase. While these salivary enzymes will generally yield the different types of carbohydrates, and that is a disaccharide. Disaccharide examples here are maltose, sucrose, and lactose will now be introduced into our small intestine, specifically in the mucosal cell. Now, what happens in the stomach is that these disaccharides will not actually be affected so much in there, because in the stomach there is only a facilitation of mechanical digestion along with those gastric juices. So basically, the most important digestion before absorption is actually happening in the small intestine. And this is happening specifically in what we call the mucosal cell. In the small intestine, where our pancreas will now secrete the digestive enzymes. And these enzymes are the following: lactase, maltase, and sucrase. All these three enzymes are actually secreted by the pancreas. What will be the function of these three enzymes? For the first enzyme, called lactase, lactase actually breaks down our lactose, a disaccharide, into a monosaccharide called galactose. The other type of enzyme, called maltase, will also break down, or maltase breaks down maltose into glucose. And finally, Sucrase. Sucrase breaks down sucrose into fructose. Galactose, glucose, and fructose, a monosaccharide, will now proceed to our bloodstream, and this is introduced via the intestinal lining called villi. This is where the absorption now of our carbohydrates happen. So remember that the galactose, glucose, and fructose are already in our bloodstream. And when they are in our bloodstream, they will be delivered to the different types of cells in our body. So how are these monosaccharides going to be converted into a more useful compound before it can be produced into an energy? And that is what we are going to talk about in here. So we will start with glucose. Glucose will undergo the metabolic pathway through a process called glycolysis. Glycolysis is a compound word that comprises of glyco, that is sugar, and lysis, that is splitting off. So in other words, the splitting of sugar. So what will be the end product for this? We will look at that later. Glycolysis happens inside the human cytoplasm, or the environment inside the cell, and there are 10 steps that will carry out the entire process of glycolysis. So again, glycolysis is actually the splitting of glucose into products called pyruvates. So the two pyruvates will be later on processed to produce high energy that will be utilized by our body processes. Aside from pyruvate, glycolysis also produces water. So now, what about fructose and galactose? Firstly, fructose, before it enters into the glycolytic process, will be phosphorylated to form a compound called fructose 1-phosphate. For galactose, on the other hand, it will be converted first to form a compound called glucose 6-phosphate. Fructose 1-phosphate and glucose 6-phosphate will now be ready to join with glucose into the process of glycolysis to produce pyruvate, a precursory compound to produce high energy for our body to use. So we will now look at the 10 steps of glycolytic reaction in the following slide. So we will now be looking at the 10 steps in glycolysis, and here it is presented in a table form. So the table is divided into 4 columns. The first column will be about the process or the steps, and the second column will be about the energy utilized, and the third column will be the controlling enzymes, and finally the last column will be about the products or the molecules that are developed. So for the first step is phosphorylation using ATP. So adenosine triphosphate is now reduced to ADP, or adenosine diphosphate, plus an ion of hydrogen that is controlled by the enzyme called hexokinase. So from our glucose, that will be converted into glucose 6-phosphate. So next step will be an isomerization. There is no energy utilized in here, but that is controlled by our enzyme called phosphoglucoisomerase. When we speak of isomerization, that's just simply converting into a different structure of our molecular formula from glucose into fructose. So that will now be converted into fructose 6-phosphate from glucose 6-phosphate. So the third step actually is phosphorylation using ATP, meaning addition of our phosphate. So again, reducing ATP into ADP plus H. That is handled by our enzyme called phosphofructokinase. So our fructose 6-phosphate will now become fructose 1, but since this is only phosphate plus one more phosphate, that will now become 6-biphosphate. For the fourth step is called cleavage, actually splitting into two molecules, and that is done by an aldolase without any energy utilization in there. And we are now going to produce two types of molecules in here. First is glyceraldehyde 3-phosphate, and the other one is dihydroxyacetone phosphate. What will we do with dihydroxyacetone phosphate? This again will be converted into another glyceraldehyde 3-phosphate, and that will happen in our isomerization, because the isomerization again will change dihydroxyacetone into glyceraldehyde 3-phosphate. So without any use of energy, because isomerization doesn't need energy to do that, it only requires a specific type of enzyme called triocephosphate isomerase. So now instead of one glyceraldehyde 3-phosphate that is happening in step 4, in step 5 we have two molecules now of glyceraldehyde 3-phosphate. And for our step 6 is now the oxidation and phosphorylation using our inorganic phosphate. So our energy that is used here is actually 2 NADH plus our 2 inorganic phosphate, and that becomes 2 NADH plus 2H. So the type of enzyme that is utilized in here is called glyceraldehyde 3-phosphate dehydrogenase. So we also again have two molecules of our 1,3-biphosphate biphosphogycerate. So that's the molecule that we are producing in here. And finally, with our number 7, phosphorylation of ADP. So from 2 ADP, that will be reduced into 2 ATP. So now there's an energy that is being produced in here, and that is done by an enzyme called phosphoglycerate kinase. So there will be two molecules of 3-phosphoglycerate. And finally, isomerization again happens in step 8 without the use of energy by the enzyme called phosphoglycerate mutase. So that will yield into a molecule or two molecules of 2-phosphoglycerate. And a dehydration in step 9, of course, we'll be making use of water for that. And the type of enzyme that is controlling that is called enolase. And so that will now be converted into, again, a molecule of phosphoenol pyruvate. So finally, in step number 10 is the phosphorylation or addition of phosphate. So our 2 ADP will now becomes 2 ATP. So pyruvate kinase is an enzyme that is utilized in here, and now we are going to produce also the molecule, precursory molecule called pyruvate, a precursory molecule for a high energy conversion. So we are producing pyruvate, we are also producing ATPs, and at the same time we are also producing water. Glycolysis pathway. Glycolysis pathway is regulated by what we call a glycolysis regulation. Glycolysis regulation is very important because if there is no regulation in the production of our molecules in the different steps in the glycolysis pathway, then it cannot proceed further to the next step. So we will be looking at the different steps wherein our glycolysis regulation can be found. So we will also see what will be the stopping point or the concentration needed so that the enzymes that do the enzymatic action will be given a signal when to stop or inhibit its enzymatic action. So our glycolysis regulation has different control points. So we will see the different control points in here. These different control points can be found in, the first one is in step 1, the glycolysis step number 1. This is inhibited by the production of glucose 6-phosphate. If glucose 6-phosphate is already created, then this will already inhibit the function of the enzyme called hexokinase, the one that also creates glucose 6-phosphate. Another type of glycolytic regulation can be found in glycolysis step number 3. Inhibition of the enzyme is done by the concentration of ATP and citrate. So when ATP is already in the highest concentration, and at the same time citrate is also present, then this will inhibit the function of another enzyme which is involved in step number 3, and that is phosphofructokinase. And finally, the last glycolysis regulation can be found in glycolysis step number 10, and that will inhibited by the concentration again of our adenosine triphosphate. So with that will also inhibit the function of an enzyme called pyruvate kinase. So these are the 3 control points where we can find the glycolysis regulation in the glycolysis pathway. So when pyruvates are already created from a process called glycolysis, pyruvates will now have different ways in converting it into an energy, and that is called the fates of pyruvate. So actually pyruvates can be utilized as an energy by the different types of organisms, and the different conditions wherein this pyruvate can be affected so much in the different steps on how they will be transformed into a form of energy. And these fates of pyruvates will actually be following the different types of conditions, and the first condition that we are going to talk here is called aerobic. So aerobic condition requires oxygen, so you have to remember that when we speak of aerobic, it has something to do with the molecule of oxygen. Now aerobic condition simply occurs in what we call the different types of organisms, such as humans, animals, and some microorganisms that also requires aerobic condition. So there are specific types of organisms in the decomposition of our trash. So the first type of organism that decomposes the trash is the anaerobic, the one that do not requires oxygen, and the last type after 15 days will be replaced by another type of organisms, the one that requires oxygen. So the aerobic decomposition and anaerobic decomposition. So in this scenario, pyruvates are being utilized in this condition to produce energy in terms of aerobic condition. And so what is the way that pyruvate can be converted into energy by means of aerobic condition? So the process is actually done in the mitochondria, and in the mitochondria they will produce acetyl-CoA. So acetyl-CoA can further be processed to produce ATP and higher forms of ATP concentration, which we will be talking in the next slide. And the other type of condition wherein the fate of pyruvate can follow is called the anaerobic condition. When we speak of anaerobic condition, again, this is a process wherein oxygen is not requirement in this step, in the different steps in the conversion of the pyruvate into energy. And so what is the process called in this step? So it's called fermentation. So fermentation is generally a term used to describe the conversion of NADH or NADH that is reduced into NAD. So Are there different types of fermentation? Well, in here, the different types are lactic acid fermentation. So, when we speak of lactic acid fermentation, this occurs in the muscles of animals, including humans. So, basically, we also utilize the other type of aerobic uh, and at the same time an aerobic condition in the utilizing of our pyruvates in the form of energy, because in our muscle and other muscles like other animals, the ATP is being produced for the energy requirement for the activity. But like athletes and the rest, this type of lactic acid fermentation will actually also build lactate in the muscle, and because of this will actually cause the muscle pain, or sometimes call it muscle soreness. So this is one type of fermentation from a anaerobic condition. Now the other type of fermentation is called alcohol fermentation. So what is the difference between lactic acid fermentation from an alcohol fermentation? Well, these usually are found in organisms such as yeast and fungus, and this pyruvate, or the carbohydrates that are converted into pyruvate, can now be transformed into a type of fermentation that will produce an alcohol. And because of this, they are very useful in the beverage industry, and also in some food like the making of the baking of bread and cake or pastry. This type of fermentation will also help to rise the dough. So they are very useful in our day-to-day life. So this is very important to understand that pyruvates actually will follow the different conditions, which is aerobic and anaerobic conditions. And so in terms of aerobic, it requires oxygen in the process to convert pyruvate into acetyl coenzyme, while in the anaerobic condition, it's just reducing our NADH into NAD. So there are two pathways still in there, either that follows lactic acid fermentation or alcohol fermentation. But in alcohol fermentation, that is mostly generally specific to organisms such as yeast and fungus, while lactic acid fermentation occurs in our muscle, especially in strenuous activities. So let's move forward to the Krebs cycle. So before we go into the different steps of the Krebs cycle, we will first look into the different concepts that will channel us and relate this connection to the concept of Krebs cycle. So again, what is cellular respiration? Cellular respiration is a process wherein our cells will convert our food into energy. So an important molecule there is called NADH or NADH. This is a potential molecule that carries an energy. So it's a chemical that generates energy. And NADH is actually produced in an intermediate phase of our glycolysis, step number 6. So NADH is produced along step number 6 in the glycolysis action, and the only thing is that NADH is not allowed to enter into the mitochondrial membrane. You see that the mitochondria or mitochondrial membrane are actually semi-permeable. It only allows a specific type of material to get inside its membrane. So since they are not permeable, but the NADH has a very important electrons that is also a requirement in the Krebs cycle in the mitochondria. So this is now carried by our molecule called dihydroxyacetone phosphate. This is a one of the products of a glycolysis. That's why it's called a glycolysis intermediate. And another molecule called glycerol 3 phosphate. So these are the ones that will bring these electrons into the mitochondria. And This energy, the conversion of food into energy, is described in what we call the Krebs cycle. So basically, it's developed by uh, a certain scientist. But is Krebs cycle the only name for it? Actually, it's called also a citric acid cycle. At the same time, it's also called a tricarboxylic cycle. So if you happen to hear these words, it is also referring to the Krebs cycle. And so who developed Krebs cycle? So it is discovered by a scientist named Hans Adolf Krebs. So the Krebs cycle is generated or generates energy by actually oxidizing the acetyl-CoA into energy. So remember that in the glycolysis, we are already producing two molecules of pyruvate. This can be further processed in the mitochondria by converting this one into acetyl-CoA. So we will be looking into the different stages of the Krebs cycle or the citric acid cycle or the tricarboxylic cycle in the next page of the presentation. But Krebs cycle happens in cytosol, meaning in the cellular environment for prokaryotic cells, such as the other types of organisms that doesn't have any nucleus. So those are prokaryotic cells. While in our for us humans, we are eukaryotic beings. So this happens inside a mitochondria. So it's located very specific inside a mitochondria. So that is for eukaryotic cellular organisms such as humans. So this is our overall summary of the Krebs cycle, and this is presented to you in table form. So we have divided the table into 5 columns with input as the first column, followed by the process and the enzyme that is involved in the process, the product or products, and the energy output. So if you can remember that pyruvate is introduced into the Krebs cycle. Pyruvate is a product of glycolysis. In the Krebs cycle, pyruvate is oxidized into a molecule called acetyl-CoA. This is done by the enzyme called pyruvate dehydrogenase complex. There are 2 products that are generated in here. One is acetyl-CoA molecule and another one is carbon dioxide. While NADH is released or produced from the process. Acetyl-CoA is our new input. So acetyl-CoA will be added with oxaloacetate to create the molecule called citrate. This is done by the controlling enzyme called citrate synthase. Citrate is a 6-carbon molecule. No output can be seen in here. Citrate and water as an input will create isomerization or isomer into an isocitrate. This is done by the enzyme called aconitase. So isocitrate molecule and carbon dioxide is the product in this process, while the output is NAD is reduced into NADH. The isocitrate and NAD+, the process will be isocitrate will now be oxidized into alpha ketoglutarate by the enzyme isocitrate dehydrogenase. Alpha ketoglutarate compound or molecule is having a 5-carbon structure because in the conversion of citrate into isocitrate, there is a release of carbon dioxide, and that carbon from the carbon dioxide is taken from isocitrate, from 6 carbon molecule now reduced to 5 carbon molecule. And in here also carbon dioxide is again released, while NAD is reduced into NADH. Alpha ketoglutarate molecule and NAD plus and CoA-SH. The alpha ketoglutarate is now converted into succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase. So the compound that is produced in here is succinyl-CoA, which is only 4 carbons that is taken from the original alpha ketoglutarate that has 5 carbons, less the carbon from the carbon dioxide that is released. Succinyl-CoA will now be having a total of 4 carbon molecule, while GTP is generated in this process. Succinyl-CoA that is having a 4 carbon molecule and GDP plus P, succinyl-CoA is converted into succinate. This is done by the enzyme called succinyl-CoA synthase. So succinate molecule is produced, while QH2 is generated that will be later on used to produce FADH2. Succinate and coenzyme Q. Succinate is converted into fumarate by the enzyme succinic dehydrogenase. Fumarate is now produced as a molecule that is a product in this process. Again, no output is released. The fumarate and water. Fumarate is converted into malate with the enzyme fumarase. Malate is the molecule that is produced in here, while NAD is reduced into NADH. And finally, malate and NAD+, malate is converted into oxaloacetate by the enzyme malate dehydrogenase. So the product is oxaloacetate with having a 4 carbon molecule. Oxaloacetate will be recycled back into the second process because oxaloacetate will create or convert acetyl-CoA into citrate, and so the process will continue to produce energy by recycling our oxaloacetate. So this is how a Krebs cycle looks like.